So the cerebrospinal fluid is a critical component of brain function. It's going to help cleanse the brain of all its waste, which according to an article just released about a week ago, appears to be a major function of the cerebrospinal fluid, is to basically flush the brain during sleep. And that's what sleep may be mostly for, is to allow the toxins that accumulate during processing and thinking all day, allow them to be cleared away during periods of lower activity at night. So the ventricles, the chambers that are found in the brain where cerebrospinal fluid is both produced and then circulated around the brain, begins with the two lateral ventricles. We'll see anatomical locations for these in just a sec. There will be one in each cerebral hemisphere. There's a third ventricle that is found just beneath the corpus callosum, a fairly small and confined space. The cerebral aqueduct, if you recall, that we noted in the midbrain slice, is an opening that allows for the cerebrospinal fluid to percolate through from the third ventricle to the fourth ventricle. And then finally, the fourth ventricle, that's fairly open space that's found between the pons and cerebellum. Once it reaches the fourth ventricle, then we can see the cerebrospinal fluid is capable of passing through an opening into the central canal of the spinal cord. Now, where is this manufactured? A good portion of it is manufactured in a region or a set of tissue called the choroid plexus. That is this spongy mass of capillaries that are easy to find in sheep brain dissections or horse brain dissections like the one you'll see in the video. There are cells co-located with the choroid plexus called ependymal cells. We discussed these in the glial portion of chapter 12, and these ependymal cells line the ventricles as well in association with the choroid plexus, and these cells produce the cerebrospinal fluid. Now, they have to be associated with the choroid plexus because cerebrospinal fluid is much like plasma inside blood, except that it doesn't have the blood proteins, and of course no cells will get out to enter and form the cerebrospinal fluid. So it's mainly a collection of glucose and electrolytes floating in water. So here we can see the lateral view of the lateral ventricles, and the two lateral ventricles, the third ventricle located just behind them, the corpus callosum is somewhere right in this region. So the third ventricle, the cerebral aqueduct that connects the third with the fourth, and then the passage on through to the central canal. Here's an anterior view, and these structures, these lateral ventricles, have been likened to appear like a ram's horns. And so we see them in brain slices here. Here's the fourth ventricle, nice little open area just anterior to the cerebellum and behind the pons. The third ventricle is an open region then beneath the corpus callosum, and then we can see different aspects of the choroid plexus inside the lateral ventricles. So here's a slice taken through the brain. This is again a transverse longitudinal slice or horizontal slice, and we can see the longitudinal fissure, but once again you see great shot of the choroid plexus here that in association with the ependymal cells will produce the cerebrospinal fluid found in these lateral ventricles. All right, there's highlighted choroid plexus. And so cerebrospinal fluid is typically clear, fills all the canals and openings. The brain is buoyant, so the brain is going to be a lot of fat, right? It's a lot of phospholipids that make up those cell membranes in the neurons and neuroglia, the oligodendrocytes that help myelinate the axons in the central nervous system. All of this adds up to a large percentage of it being fat. And so it's buoyant and it can float in this liquid of cerebrospinal fluid. We make about half a liter per day, and we filter the blood out of those choroid plexus capillaries, and the ependymal cells filter the blood to produce the cerebrospinal fluid. So it's constantly produced, and so continuously flows throughout the central nervous system. It's secreted primarily in the lateral ventricles, which then have opening into the third ventricle, so the CSF will flow into that, through the cerebral aqueduct to the fourth ventricle, and then after it passes the fourth ventricle, then it will enter into, or through a small opening, into the central canal. Now, after it circulates in the central canal, it will come back up into the brain in occupied spaces underneath the arachnoid layer, the subarachnoid space. And in that layer are going to be small projections from the arachnoid mater called arachnoid villi that serve to recapture or reabsorb the CSF, and then will supply the blood system, the cardiovascular system, with the remnants of the CSF. All right, so functions of cerebrospinal fluid, as I already mentioned, the brain is mostly fat, so this applies some buoyancy to the brain. It helps in part to keep the brain from ramming up against the side of the skull whenever we move our head slightly. So certainly lots of protection and removing waste in terms of chemical stability and supplying glucose and other necessary electrolytes and nutrients. Here is the flow pattern. We see the choroid plexus here in the lateral ventricles. Here is the third ventricle. Yes, there is indeed still some choroid plexus, so there is some production in there. We pass through the cerebral aqueduct into the fourth ventricle, where again there is more choroid plexus and ependymal cells to produce more cerebrospinal fluid, flows down the central canal, circulates around the spinal cord, comes back up the brain on both sides, and then gets reabsorbed in these little extensions of the arachnoid, off the arachnoid mater, that will then put that material back into the bloodstream. So there is an extensive blood supply. You'll cover this more when we get into A&P2, but there is so much blood supply into the brain, and there's a lot of protection for the brain from loss of blood due to, say, single vessel not supplying the correct amount of blood. The brain only occupies a couple percent of the entire weight, yet hogs an enormous amount of glucose because of its constant activity and constant usage. It also utilizes a ton of oxygen as well. So supply of blood is absolutely essential to the brain. If you lose blood for 10 seconds, you'll lose consciousness. A couple minutes, you're going to start to see some potential neural damage. And if you lose circulation to the brain for up to 4 minutes, then you're going to have permanent damage to the brain. The way we supply blood constantly to the brain is through a unique combination of vessels called the circle of Willis, so that if one of these vessels doesn't work right, another vessel can expand and deliver more blood to the brain. A unique combination that, again, you'll cover much more detail in A&P2. Okay, we also have to be careful here that we don't allow toxins and other dangerous components inside the blood from entering the brain. Thus, we've talked about this many times, blood-brain barriers due to tight junctions that exist between the epithelial cells of the cardiovascular system, and also the foot processes of astrocytes that attach to those particular blood vessels. So we always have to worry about the fact that there are areas in the brain that do not have a blood-brain barrier. There are some regions that we have to actually sample the blood in order to figure out what's in it. And in those regions, we really have to be careful about what we allow to enter the brain. So blood capillaries throughout the blood tissue we have to be concerned with, and then the capillaries of the choroid plexus as well. We have to watch those areas and make sure and maintain vigilance over any possible toxins entering the brain. So remember, tight junctions, the astrocytes with their little feet processes, and then they help also encourage the formation of tight junctions that are going to seal off and prevent easy transfer of toxins from the blood to the cerebrospinal fluid and the brain tissue. So now anything that leaves the blood system has to go through the cells, not in between them.